Hello everyone, today we'll delve into AMHR2, a receptor that has garnered significant attention within the transforming growth factor superfamily. In recent years, it has become a hot research topic for targeted therapies of various diseases, particularly in reproductive endocrine disorders and oncology. Next, we will comprehensively analyze the definition, its expression distribution, structural characteristics, signaling pathways, and the latest advances in targeted therapy. AMHR2, short for Anti-Müllerian hormone receptor, is an important member of the TGF superfamily. In recent years, it has become a research hotspot due to its crucial roles in various diseases, particularly polycystic ovary syndrome, ovarian cancer, and lung cancer. Treatment strategies primarily focus on blocking the AMHR2 signaling pathway to regulate downstream biological effects. AMHR2 expression exhibits significant spatiotemporal specificity. It is expressed in paramedullary mesenchymal cells in both sexes, disappearing in males after Müllerian duct regression. Furthermore, it is also expressed in the testes and ovaries. In the testes, expression is observed from embryonic development to puberty. The female ovary is continuously expressed from the embryonic stage to adulthood, and is mainly concentrated on the granulosa cells of preantral follicles and cavitary follicles, which provides a histological basis for its role in reproductive-related diseases. The human AMHR2 gene is located on chromosome 12 and contains 11 exons. It encodes a 573 amino acid transmembrane protein containing a signal sequence, an extracellular domain for binding AM, a transmembrane domain, and an intracellular domain with serine or threonine kinase activity. Notably, the extracellular domain structure of AMHR2 resembles that of a three-finger toxin, which is specific to TGF family type 2 receptors. AMHR2 functions through the AMHR2 signaling pathway, a complex and interconnected regulatory process. First, the ligand AM binds to AMHR2 on the cell membrane. This binding activates downstream type I receptors, such as ALK2, ALK3, or ALK6, causing phosphorylation of their kinase domains. Next, the activated type I receptors further phosphorylate SMAD1, SMAD5, and SMAD8 proteins. These phosphorylated SMAD proteins then bind to SMAD4, forming a heterotrimeric complex, which is transported into the nucleus. Finally, it binds to the promoter or enhancer regions of target genes, along with other transcription factors, thereby regulating the transcriptional expression of target genes and controlling cellular function. In targeted therapy, mirvetuximab, a humanized IgG1 monoclonal antibody, is currently attracting the most attention. It binds with high affinity to AMHR2 on tumor cells and CD16 on macrophages, thereby activating antibody-dependent cell-mediated cytotoxicity and phagocytosis and inhibiting the function of tumor-associated macrophages. This drug has entered phase I or EI clinical trials for advanced gynecological tumors and colorectal cancer. However, the phase II study was terminated early due to poor efficacy, indicating that further exploration of its mechanism and optimization of treatment strategies are still needed. In summary, AMHR2 as a receptor with important biological functions in the fields of reproduction and oncology has gradually clarified its structure, expression, and signaling pathways. Although targeted drugs such as mirvetuximab have entered clinical trials, efficacy challenges remain. Future research should further explore its mechanisms of action in different diseases and develop more precise treatment strategies. More target sharing. See you next time.